Good morning. Today we are going to talk about enzymes and how enzymes control biochemical reactions in living organisms. So, in this lesson, we are going to appreciate that biochemical reactions in living organisms must be controlled, and you are going to understand how enzymes can act as biological catalysts, and you are going to list and explain the factors that affect enzymes' activity. And the last thing, you are going to list some examples of human exploitation of enzymes. So as we said before, that metabolism means the sum of all the chemical reactions going on within a living organisms. And the main two reactions which happen in metabolism are anabolic and catabolic reactions. So, to understand the meaning of anabolic and catabolic, focus on this picture. So, the first part, you will find that in this reaction, large molecules are built up from smaller ones. And this reaction requires energy. So, this reaction called anabolic reaction. So, large molecules are built up from smaller ones by using energy. This reaction is called anabolic reaction. For example, a plant's cells use anabolism to grow new leaves or to fix torn ones. And an animal's cells use it to build the proteins that make up muscles. So here we have two examples. The first example for plant and the second one for animal. The first example, a plant's cells use anabolism to grow new leaves or to fix torn ones. And animals' cells use it to build the proteins that make up muscles. Okay, let's focus on the second part. In the second part, large molecules are broken down into smaller ones, but here energy is released. This reaction called catabolic reaction. So again, large molecules are broken down into smaller ones, but here energy is released. So we call this reaction catabolic reaction. So the main two reactions of metabolism are anabolic and catabolic reactions. Anabolic reactions, large molecules are broken down. Large molecules are built up from smaller molecules by using energy. For catabolic, large molecules are broken down into smaller ones and that will release energy too. An example of catabolic is the breakdown of food during digestion. Food is mostly made up of proteins, carbohydrates, and fats. Cells use catabolic reactions to break down those nutrients into smaller molecules. As that happens, energy is released. This energy may be used to maintain body temperature. As you see here, the breakdown of glucose, which is in carbohydrates, into carbon dioxide and water by respiration is an example of catabolism. And here, the condensation of glucose or sugars into polysaccharides is an example of anabolism. This happens in cells of the liver and skeletal muscle. So again, we have a simple example, carbohydrates, which is glucose, break down into carbon dioxide and water during catabolism reaction. And sugars, glucose, build up polysaccharides during anabolism reaction. Okay, now the question is, what determines whether glucose molecules are built up to produce, for example, polysaccharide, or broken down into carbon dioxide and water? So what determines whether glucose molecules are built up into glycogen or broken down into carbon dioxide and water? actually the answer is enzymes. So as you know before, that enzymes are biological catalysts which speed up reactions without themselves being changed by the reaction. And enzymes are proteins. Each enzyme has a special function. So some enzymes build up the molecules and other enzymes break down molecules into smaller ones. So the presence or absence of a particular enzyme controls what will happen to a particular molecules. So enzymes are proteins that function as catalysts. So here, glucose is broken down into carbon dioxide and water by using a particular enzyme. Here, glucose is called substrate, and carbon dioxide and water are called products. So again, glucose, substrate, carbon dioxide and water are products. So substrates are the molecules that react in the enzyme catalyzed reaction, So the substrates are the molecules that react in the enzyme catalyzed reaction, and the products are the molecules produced in the reaction. Okay, now let's watch this video to understand the enzymes. The enzymes more.
Enzymes are protein molecules that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, is the area where enzyme activity takes place. An enzyme works by binding to molecules, called substrates, at its active site. The job of the enzyme is to convert the substrate into a different product or products through a series of chemical reactions. Following the reactions, the products are released and the enzyme is free to act on another substrate. At the active site, the substrate should fit into the enzyme almost like a key fits into a lock. Because of this, most enzymes can fit only one substrate. To accomplish this lock and key fit, the active site undergoes a slight change in shape in order to better accommodate the substrate. This is called the induced fit model because the enzyme is induced to undergo small changes so that the substrate can achieve optimum fit. All enzymes have optimal environmental conditions that are correlated with maximum enzyme activity. Environmental conditions, such as pH and temperature, play a role in how efficient the enzyme is in conducting its reactions. When conditions are less than optimal, an enzyme will lose its configuration and slow its activity. This is due to a change in the three-dimensional shape of the enzyme and is called denaturation.
Okay, let's watch this video to understand the second mechanism of producing or how to use enzymes.
Most chemical reactions do not occur spontaneously in a cell. Instead, cells rely on proteins called enzymes to kickstart chemical reactions and speed them up, enabling cells to get the most out of the energy sources available to them. Enzymes have a unique way of kickstarting reactions. They work by binding to one or more specific molecules called reactants, or substrates. Binding occurs at a special region on the enzyme called the active site. Once the substrates bind to the active site, they form what's called an enzyme-substrate complex. As the enzyme and substrates begin to react, some of the chemical bonds in the substrates begin to weaken, causing them to link together. Eventually, the chemical reactions at the active site lead to the formation of a different molecule. This is referred to as the product. Once the reaction has occurred, the product is released from the active site. The enzyme returns to its original state and is free to react again with another set of substrates.
So, as you noticed in the video, we noticed that an enzyme is a protein folded into a complex three-dimensional shape, and this enzyme has active site. So the active site is the part of the enzyme that allows it to act as a catalyst. So if you look at this diagram, you will find that we have enzyme, and this enzyme has an active site, and we have two substrates. Now what will happen to substrate molecules when they fit into the active site? So here, substrate molecules fit exactly into the active site of the enzyme to form an enzyme-substrate complex. Now the active site brings the substrate molecules closer together, and the last stage here, the substrates now react to form a molecule of product which leaves the active site. So after moving or bringing the substrates closer together, they will produce one product. So the substrates now react to form a molecule of product which leaves the active site. Okay, if you focus in this small graph, you will find that the enzyme lowers the energy needed for the reaction. So if you compare between the reaction without enzyme and the action with enzyme, you will find that the energy needed for the reaction with enzyme is less than the energy that needs to the reaction without enzymes. So the enzyme here lowers the energy needed for the reaction, and the reaction is then much more likely to take place. So here we have the opposite process. We have one substrate. When this substrate fit exactly into the active site of the enzyme to form an enzyme-substrate complex, inside the enzyme, here the products or the substrate will break down into two products. For example, if we have a glucose and the glucose fit exactly inside the active site, so that will produce enzyme substrate complex. After that, the products will be carbon dioxide and water. Or another example, if we have lipids, the protein lipids here, enzyme is worked out as lipase. Lipase will break down lipids into fatty acids and diglycerol. So the products will be fatty acid and diglycerol. Another example, if we have a protein fit inside the enzyme, which is proteases. So proteases will produce enzyme substrate complex. After that, will divide the proteins into amino acids. So this is the mechanism of enzyme action. We called it the lock and key hypothesis. Okay, now as we said, enzymes are synthesized in living cells. So now we are going to divide enzymes into two main types. So here we divide enzymes into two main types. The first one, intracellular enzymes, and the second one, extracellular enzymes. Actually, what do we mean by intracellular or extracellular? Intracellular enzymes work inside the cell, but actually extracellular enzymes are made inside cells, then released from the cell to carry out their function out the cell. So intracellular enzymes work inside the cell. Extracellular enzymes made inside cells then released from the cell to carry out their function. Okay, now we have to know that enzymes are specific, which means that they work on one kind of substrate. Enzymes are specific means they work on one kind of substrate. So let's go back to intracellular and extracellular enzymes and let's give many examples. Let's start with intracellular enzymes. Actually, we have two examples here. The first one, catalase. Catalase breaks down harmful hydrogen peroxide in liver cells. So the first example, catalase, and the main function of this enzyme to break down harmful hydrogen peroxide in liver cells. And the second enzyme, phosphorylase. Phosphorylase actually builds glucose into starch in plant storage cells. So which enzyme builds glucose into a starch? Actually, it's phosphorylase. As we say, these are intracellular enzymes. Let's go to extracellular enzymes. Actually, we have two examples here. The first one, lipase, and the second one, amylase. Actually, lipase breaks down fats to fatty acids and glycerol. So the first example, lipase breaks down fats into fatty acids and glycerol. And the second one, amylase, which converts starch to maltose during germination. So lipase, amylase, both break down. So lipase breaks down fats to fatty acids and glycerol, and amylase converts starch to maltose during germination. So as we said also, enzymes are specific. For example, proteases break down proteins, lipase only for lipids. For example, amylase for starch. So enzymes are specific. That means they work on one kind of substrate only. One kind. Okay. Now, as we said before, again, revision, enzymes are biological catalysts which speed up the reactions inside our bodies. Now, we have to know what are the factors that affect the enzyme's activity. So, sure, we have factors. Those factors affect the enzyme activity. So the question now is, what are the factors that affect the enzyme activity? Actually, we have three main factors. The first one, temperature. The second one, pH. And the last one, activators and inhibitors. Let's start with the first one. As you know, temperature affects the activity of enzymes. As higher temperature means more active enzymes. Higher temperature, more active enzymes. As you know, that a higher temperature speeds up the movement of substrate molecules so that when they collide with the enzyme, they have more energy and are more likely to bind to the active site. But actually, all of that happened before 37 degrees Celsius. Because after 37 degrees Celsius, the enzyme activity starts to drop rapidly. This is because the high temperature starts to break some of the bonds holding the enzymes together. And so the active sites starts to change shape. So after 37 and at high temperature, start to break some of the bonds holding the enzymes together, and so the active site starts to change shape. If it changes shape too much and the enzyme won't be able to bind the substrate and catalyze the reaction anymore, and at this point would say that the enzyme has been denatured. So the temperature which an enzyme functions best is known as its optimum temperature. So here 37 is the best rate of reaction, so 37 is the optimum temperature. So the optimum temperature of this enzyme will be 37 degrees, because that the temperature at which rate peaks as the human body is kept around 37 degrees. Okay, so if you focus here, increased heat energy causes more collisions between enzyme and substrate until reaching into the optimum temperature. At the optimum temperature be the highest enzyme activity. After the optimum temperature, the activity of enzymes start dropping down. So at high temperatures, the enzyme may lose its active site, it's denatured. So here the enzyme denatured. Okay, now if you remember from chemistry, pH is a measure of acidity. So changing the acid or base conditions around the enzyme molecule change the three-dimensional shape and can denature the enzyme. So here changing the pH depending on the condition and depending on the position of the enzyme. So pH is a measure of acidity. So changing the acid or base conditions around the enzyme molecule change the three-dimensional shape and can be denature the enzyme. As each enzyme has an optimum temperature, also has its own optimum pH. For example, pepsin is an enzyme that works in the stomach and its optimum pH around pH 2. That means strong acidic. So the first example, pepsin enzyme. This enzyme works in the stomach and its pH around pH 2, which is strong acid. And the second example, amylase. Amylase works in mouth and small intestine. Its optimum pH around 7.5, which is weak basic. So those are the two examples. So let's watch this video to understand the factors that affect enzymes activity.
Hi and welcome back to freesciencelessons.co.uk. By the end of this video, you should be able to describe the effect of temperature and pH on enzyme activity. We've been looking at enzymes. We saw that enzymes speed up chemical reactions, and to do this, they've got a groove on their surface called the active site. The substrate is the molecule that the enzyme reacts with, and this fits perfectly into the active site like this. The enzyme now breaks down the substrate into the products. Remember that enzymes are specific, and that's because the substrate must fit perfectly into the active site. That's called the lock and key theory. So we're going to start by looking at the effect of temperature on the rate of an enzyme catalyzed reaction. I should point out that this is a very common exam question, so you need to learn it. Imagine I've got an enzyme catalyzed reaction, and I gradually increase the temperature. At each temperature, I measure the activity of the enzyme, in other words, the rate of the reaction. I get a graph that looks like this. As you can see, as we increase the temperature, the activity of the enzyme increases. In other words, the reaction gets faster. That's because as the temperature increases, the enzyme and the substrate are moving faster. That means there are more collisions per second between the substrate and the active site. At a certain temperature, the enzyme's working at the fastest possible rate, and that's called the optimum temperature. At this point, there's the maximum frequency of successful collisions between the substrate and the active site. For most human enzymes, the optimum temperature is 37 degrees Celsius, which is human body temperature. Now, as we increase the temperature past the optimum, then the activity of the enzyme rapidly decreases to zero. In other words, the enzyme stops working. That's because at high temperatures, the enzyme molecule vibrates, and the shape of the active site changes like this. Now the substrate no longer fits perfectly into the active site. We say that the active site is denatured, and you'll be expected to use that word in your exam. The enzyme can no longer catalyze the reaction. Okay, let's take a look now at the effective pH on enzyme activity. In this case, we're taking an enzyme-catalyzed reaction and we're altering the pH. We're then measuring the activity of the enzyme at each pH, in other words, the rate of the reaction. We get a graph that looks like this. The enzyme has an optimum pH where the activity is maximum. If we make the pH more acidic or more alkaline, then the activity drops to zero. That's because the active site denatures if the conditions are too acidic or too alkaline. Each enzyme has a specific optimum pH. So, for example, this enzyme works best at an acidic pH. This could be a protease enzyme in the stomach. This enzyme works best at an alkaline pH. This could be an enzyme released from the pancreas into the small intestine, for example, lipase. Remember, you'll find plenty of questions on the effective temperature and pH on enzymes in my revision workbook, and you can get that by clicking on the link above. Okay, so hopefully now you should be able to describe the effective temperature and pH on enzyme activity.
So, in this video, we prove that each enzyme has a specific pH and a specific temperature. So, the last factor, or the last two factors that affect the enzyme activity, actually they are activators and inhibitors. What do we mean by activators and what do we mean by inhibitors? And we are going to give two examples for both. Activators, actually they are molecules make the binding of enzyme to the substrate more likely. For example, chloride ions. So chloride ions are essential for the activity of the salivary amylase. So chloride ions are essential for the activity of amylase. So here, chloride ions work as activators because they make the binding of enzyme to the substrate more likely. And the opposite of activators are inhibitors, which make it more difficult for the enzyme to bind to the substrate. For example, cyanide ions block the active sites of enzymes involved in respiration. So the first one, chloride ions, essential for the activity of amylase, which activator. And the second one, cyanide ions, block the active sites of enzymes involved in respiration, so it's inhibitors. Okay. Now the last thing in this lesson is humans exploit enzymes. So humans can use enzymes in the field of industry, food preparation, and medicine. So here they gave a simple example, and an enzyme from snake venom can break down blood clots. So if person has blood clots inside his body, we use snake venom to break down those blood clots. Okay. Now we have finished lesson 2.5, which enzymes control biochemical reactions in living organisms. So please study the lesson, then answer the questions of the lesson. Thank you for listening and good luck.